Hello and welcome to the third episode of the Innova Biosciences webinar series. Today's webinar, How to Overcome All Your Problems with Secondary Antibodies, will introduce you to common obstacles associated with secondary antibodies in various immunodetection techniques, and will then explain the features of primary antibody labeling which helps overcome these problems. Our speaker today is Dr. Andy Lane. Andy has extensive laboratory experience in various immunodetection techniques. He joined Innova Biosciences this year, where he is well positioned to apply his knowledge to Innova's antibody labeling technologies. Today, he will introduce you to the basics of immunoassays, the properties of secondary antibodies, and explain how to overcome problems commonly associated with secondaries. There will be a question and answer session at the end of the presentation. Please submit your questions by typing in the questions box in your control panel. The questions box is circled in red here. Please feel free to send in questions throughout the webinar and we will answer as many as we can in the Q&A session. And if we do not have enough time for all the questions, we will respond to the remaining ones by email. That's the basics laid out. Now please welcome Andy.
Okay, good afternoon everyone. Thank you Claudina, and good morning to those of you joining us from the US. I'd like to start today just by thinking of some very basic fundamentals about immunoassays, leading on to talking about secondary antibodies and then how to overcome some of the difficulties that some people can associate with those in certain assay formats. Probably the most fundamental thing about an immunoassay is of course the fact that it involves an antibody. And here I've just outlined two very basic formats of an assay. A direct assay where you have an antibody binding to an antigen. This line here may be, for instance, the surface of an ELISA plate, or it could be the cell membrane of a cell. In a sandwich type assay, you're using two antibodies, a capture antibody here and a detection antibody, to initially capture and then detect your antigen. Obviously the key thing about any particular immunoassay is you need to be able to visualize those specific antibodies that are binding. To do this, you need a suitable method. That can be an enzyme if you're looking at something like an ELISA assay. It could be a fluorescent dye, or it could indeed be a nanoparticle, such as a gold particle. One of the key issues that people come across is that in many cases these reagents, these directly conjugated reagents, are simply not available. There are many tens of thousands of antigens that people may wish to detect in any particular form of immunoassay, and having directly conjugated antibodies to each of them is something that is still not reached by all of the commercial companies supplying antibodies. So a common way of overcoming this is to use a secondary antibody in a so-called indirect assay. And showing the approach here, you would have your secondary antibodies which are labeled with your enzyme or your fluorescent dye, and these will detect your antigen-specific antibody. Here in this simple direct assay, and here in the sandwich assay with your secondary antibody detecting your specific antibody. Secondary antibodies have a number of properties, and I've outlined some of the most basic ones here. The great majority of secondary antibodies are anti-species immunoglobulin reagents. So if I go back to the previous slide, you'll see that your secondary antibody is recognizing another antibody. This antibody typically might be a mouse monoclonal antibody, or it may be a polyclonal antibody, for instance, raised in rabbit. The secondary antibody will actually be detecting that species, so will be raised in a different animal. So if this is a mouse monoclonal, the secondary may well be raised in rabbit, or perhaps sheep. It would be a rabbit anti-mouse or a sheep anti-mouse immunoglobulin. A single reagent may be used in many assays. So you might be able to use one particular secondary antibody in a whole raft of assays. If you have a rabbit anti-mouse polyclonal antibody, that may be used to detect any mouse monoclonal So if you have a horseradish peroxidase conjugated secondary for use in ELISA assays, and you have a number of mouse monoclonals against different antigens, you can use the same secondary antibody in each of those assays. And that's obviously giving you some advantage in terms of simplicity for your immunoassay design. And it's also generally believed that you may get binding of multiple secondary antibodies to your specific antibody, and that may result in an increase in the signal. I'm going to talk about that in a little bit more depth later on. So those are some of the general properties of secondary antibodies that people associate with them as positives, but there are a number of challenges in using secondary antibodies. Probably the main one is that the minute you have a secondary antibody in your system, the level of non-specific binding in the assay is increased. That can come from a number of sources, but is especially important in assays where you have immunoglobulins from different species present. So a couple of examples of this shown here. We'll go back to this sandwich assay where you have your capture antibody here and your detection antibody here. Your secondary antibody, if you have your capture and detection from the same species or even a similar species, may well bind to both your detection antibody and your capture antibody. That will give you a signal even in your negative controls, and that's best seen here on the right-hand side. Here's your capture antibody. There is no antigen bound, but your secondary antibody may still recognize that capture antibody and give you a positive false signal in your assay. If we look at a similar thing in a flow cytometry or a cellular-based assay, here we have a cartoon representation of a B lymphocyte. Now, a B lymphocyte will have on its cell surface immunoglobulin, seen by these smaller antibody molecules here. We also have antigen on the surface of the B lymphocyte, and this is being detected by your primary antibody here. When you add your secondary antibodies, they bind to your primary antibody, but they will also cross-react potentially with the surface immunoglobulin. This is particularly a problem if you're using rodent systems, so perhaps these might be mouse cells and your primary antibodies are rat monoclonals, or vice versa. But you can have cross-reactivity between immunoglobulins from different species right across the species range. In your negative cell here, this is your B cell. It has surface immunoglobulin present, but none of the antigen, but you will see that the secondary antibodies will still bind. So you have a significant increase in non-specific binding and a significant difficulty in interpreting your results. In a similar way, multi-parameter assays can be very difficult to run if you're using secondary antibodies. And again, from a cartoon perspective, we can see how this will work. In this case, we have a cell which is expressing two antigens of interest, one of them in purple, another one in blue. The blue antibody is detecting the purple antigen, the green antibody the blue antigen, and you can see that this is a specific reaction. However, if you go in with a secondary antibody, you will see that in this case the secondary antibody binds both primary antibodies, and it's impossible to distinguish between those. Even if these antibodies are from different species, again, you may well see significant cross-reactivity. Now, secondary antibody cross-reactivity of this sort may be reduced in a number of ways. Species cross-reactivity can be reduced significantly by the use of antibodies that have been absorbed against immunoglobulins of the species of interest. So, for instance, if you're using a secondary antibody against mouse immunoglobulins and you don't want it to react with rat immunoglobulins, it is possible to buy a commercially available antibody that has had that absorption process undertaken. However, most of the cross-reactive antibodies are those with highest affinity. and this results in a reduction in antibody affinity and therefore performance of the antibody. So, for instance, the titer of an antibody that has been absorbed against the species immunoglobulin can be significantly reduced compared to your standard secondary antibody. Cross-reactivity, of course, can also come from other factors, such as binding to Fc receptors, and also can be due to general low-level non-specific interactions, which simply increase with the amount of antibody present. That tends to be not saturable in an assay. You have some other problems potentially with secondary antibodies as well. The increase in sensitivity that I mentioned earlier may not always be realized. The antibody binding from the primary antibody is a dynamic reaction that has an on-off situation. So antibody binding to the antigen will not always stay there permanently once it is bound. When you go through the wash steps that are involved with secondary antibodies, you can actually reduce, in some cases quite significantly, the amount of primary antibody bound to the antigen. And therefore, this is one of the reasons why you don't always see the increase in sensitivity with a secondary antibody that you might expect. And I'll show you some data a little bit later on showing exactly that in a Western blotting situation. One of the other things that frustrate people most about using secondary antibodies is the fact that your assay times are increased. The additional washes and incubation steps can significantly increase the time to run your experiment. In a direct assay, you may incubate with your primary antibody maybe for 15 minutes, perhaps up to 60 minutes, depending on the assay. If you need to then add a secondary antibody, you have to undergo wash steps, and you have to repeat the incubation probably for a similar amount of time. So your experimental time will be increased quite significantly. All of the problems with secondary antibodies that I've outlined here can fairly obviously be overcome by the use of directly conjugated primary antibodies. And going back to the same diagrams, we can see how this works. In this case, your directly conjugated primary will bind to the antigen, but it has no cross-reactivity at all to the capture antibody. In the cellular assay, your directly conjugated antibody binds to the antigen, but has no cross-reactivity to the surface immunoglobulin, and you get a much clearer result. And in multi-parameter assays, this can be seen even more clearly. Here, your primary antibody recognizing the purple antigen has a green dye attached, and the antibody binding to the blue antigen has a red dye attached. And if you were running this in a flow cytometry experiment, you could easily separate out these two colors and see which cells have which antigens on them, or indeed which ones carry both antigens simultaneously. The major problem you have with using directly conjugated antibodies isn't the fact that technically it's difficult, it's simply that in many cases they're not available. And if you have antibodies yourself, you may also struggle with traditional techniques for conjugating antibodies. This is where I'd like to introduce the lightning link conjugation technology from Innova Biosciences. These are the world's easiest to use antibody labeling kits. You can use them in a simple one-step process which literally only involves 30 seconds hands-on time. This is a highly reproducible technique and can be scaled from as low as 10 micrograms in our smallest kits up to several milligrams, or even for manufacturing purposes into tens of milligrams or higher. Very importantly, the recovery in these assays, or in these conjugations, is 100%. There are no column separation steps after the conjugation using the Innova lightning link kits, and therefore your recovery is very high indeed. In a diagrammatic form, you can see the steps here. This is your lightning link vial that is available within the kit, and we'll assume that the blue here is your fluorescent dye that you want to label your antibody to. You take your antibody, you add a modifier solution very quickly, and then add that to your lightning link vial. In this particular case, we're looking at a rapid labeling kit, and you incubate this for literally 15 minutes. You then add a quencher or a stop reagent, incubate for a further 4 minutes, and your labeled antibody is ready to use. That's a total preparation time of less than 20 minutes. The rapid kits, as I said, incubate for 15 minutes. The standard kits incubate just for 3 hours, but that still gives you plenty of time to undertake labeling and an experiment within a single day. The lightning link range comes with a whole range of different conjugation opportunities. On the left-hand side here are the whole range of fluorescent dyes that are available in lightning link format, including common dyes such as fluorescein, but also dyes such as the atto dye range and the psi dye range. We also have enzyme labeling kits for people using applications such as ELISA or Western blotting. You can conjugate to alkaline phosphatase, glucose oxidase, and horseradish peroxidase. And for the other flow cytometry applications, we can have conjugations to fluorescent proteins such as R-phycoerythrin, PerCP, and even to the tandem dyes such as PE-Psi5 and PE-Psi7. In this slide, I also would like to highlight the rapid dye-light kits that are available. So we have another series of dyes, the fairly well-known dye-light dyes that are available in the rapid kit format, further extending the range of products available in the lightning link range. There are some considerations that you need to be aware of in terms of conjugation. The lightning link conjugations are very, very simple indeed, but you do need antibody in the right format for them to work most effectively. Commercially available antibodies come in many forms, and it's best to check with the supplier about some details. Most of those details will be available on a product data sheet, but the things that you need to look for are as follows. Firstly, the concentration needs to be at around about 1 milligram per mil or higher. You also need to check on the purity. Ensure that the antibody has actually been purified, and for instance you don't have a preparation which is simply a CITES fluid or tissue culture supernatant. So ensure that any contaminating proteins such as albumin have been removed. And as I mentioned here, make sure they haven't been put back in again afterwards. That might seem to be quite a strange remark, but in many cases antibody manufacturers may add protein stabilizers such as bovine albumin back into a purified antibody, and these can interfere in some cases with the labeling reaction. Also make sure that your buffer formulation is suitable. Most common formulations such as a phosphate buffered saline will be absolutely fine, but ensure that any amines such as glycine are truly absent and that there aren't any thiols such as DTT or mercaptoethanol present. Tris buffered saline with tris up to levels of about 20 millimolar is absolutely fine. Now, if you have an antibody but it doesn't meet these criteria, don't worry about that. You can use a purification kit to purify, concentrate, or even simply change the buffer that your antibody is in. And Innova provides some very simple purification kits for those purposes. On our website you will find this diagram which really outlines how you can approach the purification stage. So you can basically ask a number of questions. If your antibody is not purified and you know what the starting material is, you can work your way through to determining which purification kit you want to use. If you have a purified antibody but you have a contaminant that you need to remove, again you can follow the yes-no answers here to determine which of our purification products will work best for you. These are all very simple to use purification systems. You don't need any special equipment. If you have a centrifuge, that will be absolutely everything that you need to undertake this. So once you've made your new conjugates, there are a number of things to consider. Basically, you will use them exactly as you would with a normal staining technique. One of the things, though, to be aware of is that you need to titrate the conjugates and possibly quite extensively in your assay system. If you remember, you will have conjugated your antibody at around about 1 milligram per mil. That's a very high concentration of antibody for use in immunoassay, where antibodies would normally be used perhaps at 1 to 10 micrograms per mil. So you will need to test your antibody at a range of dilutions to make sure that you get the best positive staining whilst minimizing any non-specific staining simply because you have high concentrations of antibody. Storage of the antibody is probably best at 4 degrees centigrade in concentrated form. So it's best not to dilute your antibody. Say if you do the titration and you find you can use it at 1 in 1,000, don't dilute everything 1 in 1,000. Keep your stock solution in concentrated form and dilute your antibody ready for use. A preservative would be useful, such as sodium azide. And if you do store any of the antibody in a diluted format, a carrier protein such as bovine albumin would be valuable to use. It's possible to freeze some of the conjugates, but others should not be frozen. For instance, you should never freeze conjugates of phycoerythrin, allophycocyanin, or any of the tandem conjugates. If in doubt, it's best not to freeze a conjugate, but to keep it at 4 degrees. Fluorescent conjugates are best kept away from light. Tandem dyes are particularly sensitive to light. I'd like to just finish off with some example data looking at the differences between using directly labeled antibodies and indirectly labeled antibodies. And I've used 2 Western blotting examples to illustrate this. In this diagram, there's been a Western blot undertaken using an antibody against an antigen called quinone reductase 1, which is expressed in human kidney. In the left-hand column, this antibody has been directly labeled using a lightning link kit to horseradish peroxidase. And the primary antibody has been applied at 0.004 micrograms per mil. After the blotting has been completed, an exposure time of 10 minutes has been used, and you can see the band at around about 30 kilodaltons of this antigen being very clearly expressed. Exactly the same antibody has then also been used, but in a typical 2-stage secondary antibody approach. Here the primary antibody has been used actually at nearly 25 times higher concentration, and a secondary antibody has been applied afterwards. So you've had to undergo all of those wash steps and the additional time taken for the secondary antibody to be applied. And what you can see, even with 25 times more primary antibody, is the band seen is really of exactly the same intensity. There is no additional intensity seen using that antibody, even though you're using much more of the primary reagent. In this second case, again, using a different antigen but the same approach, you can see that the antigen called GFAP, which is expressed in mouse brain, here this antibody has been used in a directly conjugated format at just under 0.2 micrograms per mil, and very clearly you get a Western blot positive band at around about 45 kilodaltons. If you use the same antibody at 0.5 micrograms per mil, so in this case 2.5 times as much primary antibody, you will see a band in a similar place. It's a similar intensity, but it's possibly not quite as clear in terms of the binding that you get. So once again, you can use less primary antibody, you can save the time for the secondary antibodies and all of those wash steps and get results that are just as clear. I'm now just going to pass back briefly to Claudina to talk about how we'll handle the question and answer session and a couple of other things that what Innova are up to at the moment.
Thank you, Andy. It's time for our Q&A session now. Thank you for sending in your questions. If you still have questions pop into your head during the Q&A session, please feel free to type them into the questions box, as we will reply to the remaining ones by email. And we also welcome any other feedback that you may have. We're also going to be exhibiting at the American Society for Cell Biology annual meeting in San Francisco in a couple of weeks. That's from December 15th to the 19th. If you're also going to this conference, please contact us to set up a meeting, or remember to drop by booth 442 and say hello, as we will then be able to answer any further questions that you may have about direct antibody labeling. And we will also have an exciting new giveaway with us, the Innova Biosciences Fluorochrome Slinky, which you can see on the picture here. And if you come by our booth, number 442, you will receive one of these. Please bear in mind that this webinar is recorded, and we will upload it to our YouTube channel in the next couple of days so that you can always come back to it. You can find our channel at youtube.com/Innova Biosciences. Please keep an eye out on our social media channels, as we will soon be advertising our webinars scheduled for 2013. And if you would like to receive a PDF copy of the presentation or a link to the video recording of the webinar, please contact me. You can find my contact details on any of the webinar emails you've received so far. All right, let's move on to the questions then.
Great. Thank you, Claudina. We have, I think, a number of questions that are, some of them overlap, and so we'll probably try to answer those together. I think if we start with a question from Cheryl Watson, which is, how stable is the antibody labeling? Can you conjugate and use the preparation for some time after that? And yes, the antibody labeling undertaken with the lightning link kits is a very stable reaction. It's a covalent bond, and therefore you basically have a permanent link between the antibody and either the enzyme or the fluorescent dye you'll be using. So in terms of keeping that product, if you use basically the hints that I gave you in terms of storage, you can keep the antibody for a considerable time after conjugation. Actual stability will probably depend very much on the antibody itself, but you'd certainly expect it to be a year, perhaps 2 years in most cases.
Okay, another question that's come in is, are there differences in the strength of the fluorescence emitted between, for example, ATO488 and dye-light 488?
Okay, so in relation to the intensity, certainly between those two dyes, there's probably little difference in intensity. If you have dyes or an antigen, for instance, that you're trying to detect and it's particularly weakly expressed, perhaps in a flow cytometry situation, then some dyes tend to be significantly brighter than others. If you have a weakly expressed dye, I would tend to use something like picoreferin as the antibody or the conjugate of choice. But certainly between ATO488 and dye-light 488, I'd expect a similar level of staining.
Okay, the next question is there still the possibility of nonspecific binding?
Okay, so in relation to nonspecific binding, you certainly won't find the similar causes of nonspecific binding that we talked about in terms of secondary antibodies. So obviously you won't have cross-species binding due to cross-reactivity between different immunoglobulins. However, in any immunoassay, there are certain types of non-specific binding that can take place. One of these may be via the FC receptors that may be present on certain cells in flow cytometry. So for instance, monocytes are a cell type that will express FC receptors, and if your primary antibody is of an isotype that is going to bind to those FC receptors, you may see that binding. You can control for that with a relevant isotype control, i.e. using an antibody that is not specific for your antigens, but is of the same isotype and conjugated to the same dye. Okay.
The next question is the number of the conjugated sites the same in antibodies from various sources?
Okay, so I think I've got the question right there, thinking that we're talking about perhaps different species of antibodies. So for instance, a monoclonal raised in mouse or rat, or perhaps a polyclonal antibody. And in general terms, the answer will be yes, although the fine structure of an antibody may differ slightly. So it's probably one of those questions that is impossible to answer absolutely, but you would certainly expect the labeling to be similar between antibodies from different species.
The next question is it possible to label Fab, fragments, or recombinant antibodies?
Okay, so in terms of antibody labeling, or indeed protein labeling as a whole, the lightning link kits can be used to label effectively any protein that has a free amine group available. So Fab fragments may be used for labeling, recombinant antibodies may be used for labeling, and the only thing you need to be aware of if you're labeling either different proteins or antibody fragments is that the kits are generally designed, and the protocols that are available with them are designed for whole IgG, which tends to have a molecular weight of around about 150 kilodaltons. If, for instance, you were going to use a Fab2 fragment, that would have a molecular weight of 100 kilodaltons, and you would adjust the amount of antibody that you put in to the kit so that you end up with the same molar ratio at the end of the process. So yes, you can label Fab fragments, recombinant antibodies, or indeed a range of other proteins that have amine groups available.
The next question is it possible to label antibodies with gold nanoparticles?
Yes, indeed. We've focused perhaps here on kits for labeling with fluorescence particles, or shall we say fluorescent dyes, and indeed maybe with enzymes such as horseradish peroxidase. Innova also has available a range of gold nanoparticle labeling kits using our InnovaCoat binding technology, and details of those are also available on the website.
The next question is there a kit available for labeling antibodies with oligos?
Okay, that's an interesting question. Labeling antibodies with oligonucleotides is a relatively new technology that's been developed in the last few years. And Innova Bioscience have a considerable amount of experience in actually labeling antibodies with oligonucleotides. We don't currently have available a kit for this, but we do offer custom services for labeling antibodies with oligonucleotides, and again, details are available on our website, and if anybody wanted to contact us about that, we can provide you with details of those services that we can offer.
The next question is, once labeled, how much change is there in the working titer?
Okay, that's also an interesting question. Again, it will depend on the technique that you're using. So, for instance, if you were using an ELISA technique and you've been used to using a secondary antibody system in there, you will probably be used to using your detection antibody at a certain concentration, perhaps maybe 1 to 5 micrograms per mil. Based on results we've seen, you can probably use a similar concentration with your directly labeled antibody. And as you saw with the Western blotting data, indeed in those cases you could use less antibody, i.e. antibody at a lower concentration than people had been using with secondary antibodies. So effectively, you probably wouldn't expect much change in the working titer, but you would have to test that out in your own system to be certain.
Okay, our next question is, can you vary the loading of antibody on the label when using the Lightning Link kits?
Okay, that's another interesting question. As I mentioned earlier when we were talking about the antibody fragments, I think the kits are designed for using with whole immunoglobulin, and we give you recommendations for the amount of antibody that can be used. But if, for instance, you were to look at the protocol for our horseradish peroxidase labeling kits, we actually give details there of how you can slightly adjust the ratio of the enzyme to the antibody. And it's possible basically by adding different amounts of antibody to get more enzyme bound to that antibody, or indeed less, and that may help a particular assay to gain more sensitivity.
Okay, well, I'm afraid we're running out of time now, so that's the end of the question and answer session and the webinar. But thank you for joining us today. Please bear in mind that the recording will be available soon, as well as a PDF copy of the presentation. So if you would like either, please do contact me. And we hope that you enjoyed our webinar today. Thank you.
Great. Thank you, Claudina. And I would just add, for a couple of questions we haven't answered, we'll get back to people by email very quickly. Thank you, everyone.